Welcome to this Metrion Biosciences webinar. The title of today's webinar is Recent Progress Towards a Potential Treatment for KCNC1-related Disorders. My name is Chris Mathis. I'm the Chief Commercial Officer at Metrion Biosciences. We are a specialist preclinical CRO specializing in ion channel research since 2015. With Metrion, you can access cell lines. We do high throughput screening, assay development. We have SAR support. In the cardiac safety realm, we can do GLP-hERG testing, FIFA panels, human stem cell-derived cardiomyocytes, phenotypic cardiac assays, many neuroscience assays. Many of you may not know that Metrion is a specialist in CNS and neuronal assays, current and voltage clamp, rodent ERGs, neuroscience translational assays. We also do integrated drug discovery. So without further ado, I'd like to introduce our first speaker and also just welcome everyone that's joining. We have a diverse audience today. This is a unique opportunity for us to have presenters that represent the patient population, the medical, clinical aspect of research, and then the preclinical kind of reductionist approach to the research with Metrion Biosciences. So I'd like to have a special welcome to the parents of patients that are joining the webinar today. So our first speaker is Stephanie Teleska. She is the co-founder of the KCNC1 Foundation. She is the mother to Eliana and a clinical prenatal genetic counselor. Eliana is now 3 years old. She was diagnosed at 9 months with V434L in the KCNC1 gene associated with the KCNC1 developmental and epileptic encephalopathy. Following her daughter's diagnosis, she and her husband established the only KCNC1 Foundation and promptly began research towards a treatment, connecting with researchers, CROs, and families worldwide with the aim of providing a brighter future for all those affected with KCNC1-related disorders.
Thank you, Chris, for the introduction. Good morning and good afternoon to all, depending on your time zone. And thank you for taking the time to listen to this webinar so thoughtfully arranged by Metrion. As mentioned, I'm Stephanie, the co-founder of the KCNC1 Foundation. And for just over a little of a year now, we have been working closely with Metrion on this very personal and exciting project. A special thanks to Perlara, who connected us with Metrion, and the team of scientists working with us. Gary, Scott, for all your support and guidance. I'm going to take a few minutes to introduce you all to my story and to the KCNC1 community before we jump into the rest of the webinar. So in 2021, my husband and I welcomed our baby girl into this world. We were thrilled and overwhelmed. Our path to a diagnosis is not unlike many others who walk this path. Shortly after she was born, she was very irritable and she cried so much. We did not know why, and we started seeing different specialists like a craniosacral therapist. We went in to see a pediatrician before she was supposed to see one. We went to like an urgent care center, and there they gave us lansoprazole in case she had silent reflux. Nothing was helping, but by the time she was 5 or 6 weeks, things were improving. The crying subsided, but at that time we noticed that her eyes were not moving. Typically, they were roving eye movements. We also noticed a lack of a social smile and no eye contact. At that time, panic started to set in, and our journey to her rare disease began. At 9 months, she was diagnosed with failing 434 leucine, likely pathogenic variant in the KCNC1 gene. The geneticist sharing the results gave us a generic run-through of what it could be associated with, like a progressive myoclonic epilepsy with cognitive decline, hypotonia, intellectual disability, but he just was not really sure what we could expect, and there wasn't sufficient information to share. To be honest, I was relieved to receive a diagnosis. I knew there wasn't a chance that this was just typical development, so a quote-unquote normal whole genome sequencing result would not tamper our worry. A diagnosis gave us the power to help Eliana, and we started to search for patient groups or a foundation, something to tell us that there was a hope for a brighter future. But this was the farthest from the truth. We quickly found out that there wasn't any organization uniting patients affected by KCNC1 variants or fighting for treatment. We then connected with Dr. Goldberg and learned that he was really the only academic actively studying KCNC1 and its associations with human diseases. So this is really how the KCNC1 Foundation was born. We are concurrently supporting research for a treatment alongside Dr. Goldberg. There are many variants in the KCNC1 gene with different associated phenotypes. It will not be a one-size-fits-all treatment, and we're tackling the variants one by one, so to speak. So the KCNC1 Foundation started with Eliana. We now have 39 other patients in our online support group with 14 different variants. Some cause a gain of function, some cause a loss of function. There's one variant with the dominant negative impact, and many we do not have functional studies to clarify what their effect on the channel is. Many children have seizures, many do not. Many children walk, some do not. Many children have difficulty with speech, many have intellectual disability. There's a lot that remains unknown. Eliana is now 3.5 years old. She is far from the typical toddler. She does not present like the other children in our disease group. She is non-mobile. She cannot communicate with us. She does not self-feed. She requires full assistance with every task. Her cortical vision impairment is severe. She does not recognize our faces. She does not light up when she sees our dog. We cannot make her laugh by making funny faces. But we know no difference. She is our child and we love her fiercely. We want only the best for her, to give her a chance at autonomy and to be an active participant in her life. And I think really this is what any parent would want for their child. It just looks a little different for us on how we do that for her. So now I just want you to meet the rest of our patients. As I mentioned, we've united patients worldwide, and we've built an online community for those who want to connect with each other, and we hope to continue connecting patients and their families. Now I'm going to take some time to introduce you to the faces and voices of the KCNC1 community and share with you some experiences parents have on raising a child with a rare disease. And this is all going to be shared with permission from the parent and where appropriate the child. Having children with special needs can be challenging. Getting people to understand can be the hardest part. It's the little things that make her happy, but we always need to be 5 steps ahead of her. We have too little time for our oldest kid. Our life did not turn out as we wished. It's frustrating not knowing what may come. Having a child with a rare disorder has been heartbreaking, frustrating, and isolating. The only treatment available has been medication to control seizures. Nothing has worked. We're excited that there is work being done to help kids like my son. I hope it comes soon. Yes, it was hard not knowing the why. He's the kind of guy who makes you stop and smell the flowers. The little things are big things. If all that ever comes from a diagnosis is being connected to another parent and giving them insight into their child's life is what I get, then it's all worth it. Katie is full of sunshine. Her condition and its progression have a massive impact on her life and that of the whole family. She is now age 14 and uses a wheelchair. It's a lonely space, and we're hopeful that medical science can bring some form of treatment. It was truly a shock, but we also felt a sense of, okay, we have the answer. Now what? Let's get working. Let's do anything and everything for her. She's in physical and speech therapy. Her seizures are well managed with Kv3 twice a day, and we hope she continues to amaze us. I feel so lucky for my family, but it comes with many heartaches of the unknown, wanting answers or fixes. It's hard to watch your children suffer. We love her so much. It's been so hard, but lately it's felt like we're finally just living. And so the goals of our foundation primarily revolve around accelerating research towards a viable treatment for those with KCNC1 disorders. And we recognize the complexity of this goal and will begin with identifying treatment for the V434L in KCNC1 variant. Secondarily, our goal is to unite patients and their families affected by KCNC1 disorders, support newly diagnosed families, and help provide hope when they receive this life-altering diagnosis. Additionally, by uniting patients, we will also enable the development of a patient registry. Lastly, we know KCNC1's involvement in human disease is relatively new, and basic research to understanding all the ways pathogenic variants in this gene impact an individual is not fully understood. Supporting basic research remains a goal of ours. So if you'd like to connect and/or support us, here are some ways to do this. We are a 501c3 in the USA under fiscal sponsorship of the Rare Village, and we also have a GoFundMe for those who may be interested in donating to our foundation. If you or anyone you know may be interested in collaborations, please reach out directly to me via email. Thank you all for your time.
Thank you very much, Stephanie, for that heartfelt presentation and introduction. It really puts everything in perspective. And I really admire your resolve and determination to set up this foundation, and we appreciate you presenting today. So it's my pleasure to introduce Dr. Ethan Goldberg. He's an associate professor in the Division of Neurology at the Children's Hospital of Philadelphia and in the Departments of Neurology and Neuroscience at the University of Pennsylvania Perelman School of Medicine in Philadelphia. Dr. Goldberg did his MD and PhD in neuroscience from NYU, where he trained in the laboratory of Bernardo Rudy, and general pediatric medicine, pediatric neurology, adult neurology, and neurogenetics at the Children's Hospital of Philadelphia and the University of Pennsylvania, with experimental epilepsy training in the laboratory of Douglas Coulter. Dr. Goldberg is attending physician and director of the Epilepsy Neurogenetics Initiative at CHOP, the largest dedicated epilepsy neurogenetics program in the United States. He specializes in the diagnosis and care of children with complex neurological disorders, including epilepsy, developmental delay, intellectual disability, and autism spectrum disorder. His biomedical research lab studies basic mechanisms of childhood epilepsy and other neurodevelopmental disorders, with a particular interest in ion channelopathies and how genetic variation in ion channel genes leads to human disease. His lab uses a range of approaches, including human and mouse genetics, ion channel, cellular and synaptic neurophysiology, pharmacology, induced pluripotent stem cell technology, cell-based therapy, behavior, computational modeling, and imaging approaches, including voltage and imaging and two-photon calcium imaging in awake behaving experimental animals in vivo. Dr. Goldberg will also serve as director of the BRIDGE, Brain Research in Development, Genetics, and Engineering Center of Emphasis at the new Morgan Center for Research and Innovation at the Children's Hospital of Philadelphia, expected to open in September of 2025. Dr. Goldberg is recipient of numerous awards, including the Burroughs Wellcome Fund Career Award for Medical Scientists, American Society for Clinical Investigation Young Physician Scientist Award, the Dana Foundation David Mahoney Neuroimaging Award, March of Dimes Basil O'Connor Research Award, the Grass Foundation Award Neuroscience, and the Fritz Dreyfuss Award from the American Epilepsy Society, among others. Dr. Goldberg is committed to the education of diverse learners across levels of training from undergraduate to junior faculty to the care of children and their families affected by neurodevelopmental disorders, and to the research and development of disease mechanisms, therapies, and cures. His presentation today, as you can see, is entitled Models and Mechanisms of KCNC1-related Disorders.
Thank you very much. So I did want to mention that part of the work that I'm going to be showing today was partially supported by Tiffany Therapeutics. So as was mentioned, the goals of my research program are to understand how genetic variation underlies neurologic disease with a particular focus on ion channelopathies, and I'll explain why that is. as well as neurodevelopmental disorders linking from gene, in this case KCNC1, to protein function, Kv3.1, how these genetic variants influence how neurons function, how networks and connected circuits function, and how that leads to behavior and abnormal behavior, including developmental delay, intellectual disability, autism spectrum disorder, and epilepsy, and then using that information to guide therapy. So just a little bit of background. This isn't exactly a talk about epilepsy because KCNC1 related disorders are about more than epilepsy, but it's a good starting point. So we all know what epilepsy is. It's defined by spontaneous recurrent seizures, which have an electrical basis. Epilepsy is very common. So about 1.2% of the entire United States population has active epilepsy at any one time, which is over 3 million people nationwide, which includes about 500,000 children. So according to the latest estimates, of about 0.6% of children have active epilepsy. So if you think about your child or your grandchild, if they have 500 kids in their class, about 3 of those children have active epilepsy. So epilepsy is one of the most common chronic disorders of childhood, not just chronic neurologic disorder, but chronic disorder of childhood overall. And it's associated with a markedly increased mortality rate, as well as a range of other associated comorbidities like intellectual disability, autism, etc. Epilepsy has diverse causes, but a major component, particularly in children, is genetic variation. So at least half of childhood epilepsy has a genetic cause or major component. And it's important to note, particularly in this context, that these anti-seizure medications target symptoms, not the underlying disease mechanism. So I explain this to students as going to the doctor and being diagnosed with pneumonia and being prescribed a cough suppressant. What you want, of course, is an antibiotic that treats the underlying disease, not a symptom of the disease. So a large component of epilepsy is associated with treatment resistance, and that's particularly the case for some of the genetic epilepsies that we're going to be discussing. So here's a famous slide on the left showing the various causes of epilepsy, and there are acquired causes that and are different colored slices of the pie there. Birth injury, infection, traumatic brain injury, brain tumor, Alzheimer's disease, etc. And then everything in blue, which is about 2/3 of the cases, are genetic. Now that could be even some of the acquired causes may have a genetic predisposition. So not everyone with a traumatic brain injury develops epilepsy. Now the monogenic epilepsies, the single gene epilepsies, probably constitute somewhere in the 3 to 5% range of all cases. This is an old slide, but this is our initial epilepsy panel on the right, and you can see I've colored the ion channels and neurotransmitter receptors orange and green respectively, so we can see that these types of genes are overrepresented as genetic causes of epilepsy. Now, at this point, there are over 1,000 genes, and I saw a number recently of 7,000 genes that are in some way associated with epilepsy. So there are many and diverse genetic causes of epilepsy, and de novo genetic variation is a major mechanism. So in this case, we have unaffected parents, and they have a child who can be affected with a severe epilepsy. And pardon me, I'll just hide this. There we go. So the origin of these heterozygous de novo variants presumably is one germ cell from mom or dad, or a spontaneous variant that arises after conception. So these are genetic, but they're not directly inherited per se. And I mentioned there's this disorder is defined by genetic heterogeneity, with no single gene accounting for a large proportion of cases. So to combat this issue, in 2019, we established the Epilepsy Neurogenetics Initiative, as was mentioned. So previously, we would really only have the capacity to see and offer advanced neurogenetic testing for the developmental and epileptic encephalopathies, the most severe forms of epilepsy. Whereas now, I don't have time to go into this in great detail, but we have a multidisciplinary clinic with 10 or so neurogeneticists, 8 genetic counselors, and then we offer enrollment in research studies, and we performed over 2,500 neurogenetic evaluations for epilepsy last year, offering access to advanced neurogenetic testing and research enrollment to any child with epilepsy, appropriately selected. And then that drives our research. So here's an example here of a KCNC1 related variant that we're studying. Patients have access to enrollment in research. We characterize variants at the ion channel level in heterologous systems. We obtain biosamples for generation of stem cells to turn into neurons in 2D culture and then in 3D culture, so neuronal organoids. And then for selected cases, we can generate mouse models for developing translational therapeutics. So this slide, this is actually an old slide, but this shows both the diagnoses that we've made in our epilepsy neurogenetics clinic, and you can see SCN1A, which encodes the voltage-gated sodium channel subunit NAV1.1, the cause of Dravet syndrome, which is the most common genetic epilepsy. Is top on the list, and it also shows the genes that my lab is working on, and you can see KCNC1 there. So again, this just highlights the theme that the top 10 genes on this list account for probably 75% of the diagnoses, and there's an overrepresentation of ion channels. So 7 of these top 10 genes are ion channels, and 3 of them are potassium channels. So what's a potassium channel? So the human genome contains over 90 potassium channel genes, and at least 20 of these are associated with human disease. So why is that? Well, we think that the diversity of potassium channels confers flexibility and functionality to the hundreds of different types of neurons that exist in the brain. And these potassium channel genes are grouped into these families and subfamilies based on DNA sequence similarity. And let's see if I can bring up a pointer here. Yes, I can. So here's Kv3, which has 4 subfamily members, Kv3.1 through Kv3.4. So KCNC1, the gene that we're talking about today, encodes a subfamily member of this Kv3 group, which is a member of Kv, the largest family of potassium channels, the voltage-gated potassium channels. This was the first study showing that KCNC1 was a human disease gene, and this single recurrent variant causes a disease called progressive myoclonus epilepsy, which presents first typically with myoclonus, or thought to be tremor. Average age of onset is 10 years with a range, as you can see here, and that myoclonus is progressive. Patients develop ataxia, they lose functional limb use, and then it's associated with autosomal dominant inheritance. Most of the cases are de novo, but some of them with a later onset are inherited from a affected parent, and it's associated with normal cognition or some mild cognitive decline, and it's phenotypically identical to the best known progressive myoclonus epilepsy, Unverricht-Lundborg disease. So what are the progressive myoclonus epilepsies, just to diverge a little bit? So these are a collection of rare but severe progressive neurologic disorders that are defined by myoclonus, tremor, ataxia, epilepsy with onset in a previously normal child or young adult, with relentless neurologic deterioration leading to loss of ambulation, severe disability, and in many cases early death. So treatment is symptomatic and isn't mechanistically oriented towards the underlying genetic deficit. So these treatments are typically minimally effective in controlling seizures, myoclonus, and ataxia, and have no influence on long-term disease course. So here are all the progressive myoclonus epilepsies, and I'm not going to go through all these, but I'm just highlighting that Unverricht-Lundborg disease at the top in purple, and then progressive myoclonus epilepsy type 7 due to KCNC1 variant, this recurrent arginine 320 histidine in pink. And this is the first progressive myoclonus epilepsy due to variants in an ion channel alpha subunit. So this has an interesting history. Just to mention it very quickly. So, Unverricht was a German neurologist and physician who actually has many diseases named after him, including Unverricht's fistula, which was a tubercular pneumothorax. He discovered and first described dermatomyositis, and he also described two families with what he called familial myoclony or familial myoclonus, which he described as a pure type of progressive myoclonus epilepsy that didn't really have any other major symptoms other than myoclonus and epilepsy. So the concept of the progressive myoclonus epilepsy was subsequently introduced by the Swedish neurologist Herman Lundborg around the turn of the century on the basis of several Swedish families with a common ancestor who had markers of degeneration and a form of epilepsy associated with progressive myoclonus. So unfortunately, the story of Lundborg is a sad one, as his interest in genetics led him to found the notorious State Institute of Racial Biology in 1922, and he came under disrepute because of his due to his adherence to Nazi ideology and his advocacy of eugenics and the sterilization of quote-unquote genetically unworthy individuals. So here we have some insights into how variants in Kv3.1 lead to disease from the recent cryo-EM structure. So on the left, here we have the Kv3.1 protomer in a cartoon representation. So the C alpha atoms that are implicated in disease are shown as spheres here. And then on the right, in cartoon representation, we have the voltage sensing domains S1 through S4, although S3 is omitted here for clarity, in Kv3.1A, showing the relative positioning of the gating charges R1 through R4 here. So the channel has 4 positively charged arginines that are important for opening and closing of the channel in response to changes in voltage. And that's relative to these conserved residues, phenylalanine 256 and glutamate 249, which you can see here, of the hydrophobic charge transfer center, which is located in S2. So what's unique about Kv3 channels that I'll go into further in a second is consistent with this high activation potential of Kv3.1, the S4 helix of the voltage sensing domain is in a less activated conformation compared to other voltage gated potassium channels like Kv1.2, Kv2s, Kv4s, because only 3 of these 4 positively charged arginines of the 4 voltage sensing arginines are located above the conserved phenylalanine 256 and S2, which is again part of this hydrophobic charge transfer center defined in previous Kv channel structures. So this S4 sensor here is shifted downwards by 1/3 of a helical turn compared to other potassium channels that have a more negative voltage dependence of activation. Okay, so here this tells us a little bit more about Kv3 channels. So this is some in situ data from RECAN, which shows its developmental expression pattern in mouse, and it's not really present at postnatal day 7, and then its expression increases developmentally with high levels of expression in the granule cell layer in the cerebellum, the nucleus reticularis thalami, as well as scattered neurons in the neocortex and hippocampus and this is RNA-seq data from Alan Brain showing that it's enriched particularly in GABAergic inhibitory interneurons that express parvalbumin. These are called fast spiking interneurons, and that's because Kv3 current underlies the ability of diverse neurons to fire at high frequency. So how does it do that? Well, in black, this is actually a figure from my PhD thesis that I made. So in black, we have an action potential, and what we can see is that the Kv3 current, because of its positive shift in voltage dependence, it has a uniquely positive shift in its voltage dependence of activation. It doesn't quote unquote turn on until the peak of the action potential, but it activates very rapidly and drives repolarization of the spike. Then it deactivates so that it doesn't contaminate the interspike interval, which allows the cell to fire again. And those combination of features underlie the ability of Kv3 channels to facilitate high frequency firing. Okay, so now in part 2 of the talk, I'm going to talk a little bit about our mouse model of progressive myoclonus epilepsy type 7. So this is work from Jerome Cloteau. These are families of voltage-gated potassium currents. This is Kv3 current. It's a delayed rectifier current. So you can see that when we can introduce the channel into a heterologous cell. This is a mammalian cell that doesn't express any other ion channels. So what we record is what we put there. Here's wild type Kv3. As we depolarize the cell, we get these currents that we can plot here. And here's our arginine 320 histidine. And as opposed to initial data obtained in Xenopus oocytes, it's actually about a 50% loss of function with a partial dominant negative action. And then here's the recurrent variant, this alanine 421 valine, which as opposed to being associated with progressive myoclonus epilepsy, is associated, as mentioned by Stephanie, with developmental and epileptic encephalopathy. And that's an almost complete loss of function here. There's no current. Okay, which makes sense, right? Because progressive myoclonus epilepsy is a terrible disease, but it has a later onset. It has more limited effects on cognition, whereas this variant, recurrent variant, is associated with a very severe neurodevelopmental syndrome. So that fits, so to speak. Okay, so we made this mouse, and just to sort of stick to time, you could see that the heterozygous mice are normal size, and they have normal survival. The homozygous mutant mice have early mortality due to severe epilepsy. Their brains are normal size all the way out to 6 months, but they have early evidence of cerebellar dysfunction with hind limb clasping and then progressive impairment on various assays of cerebellar function, including rotor-rod, elevated beam walking, ledge test, etc. They have audiogenic seizures pre-weaning, and then they have accelerated pentylene tetrazole kindling, and then rare spontaneous seizures that we see starting around 4 months. So it's actually quite a good phenotype of the human condition, with the prominent movement disorder and more mild epilepsy with this sort of progressive and later onset course. So here we're recording from granule cells in the cerebellum, and in both the wild type, the het, and the homozygote, and I don't have time to go into it, but basically we see abnormalities specifically consistent with Kv3 dysfunction. Interestingly, we see this before the mice are symptomatic, but those spikes are wider, and the firing frequency of the granule cells is lower, but we don't see any effects on passive membrane properties like resting membrane potential or input resistance. And then same thing in these fast spiking GABAergic interneurons in neocortex. We see some mild impairments early that become worse at later time points, and very profound abnormalities in the homozygous mice, including broader spikes, lower maximal steady state firing frequency, attenuated fast deep after hyperpolarization, etc. So we collaborated with Tiffany Therapeutics, who has developed a class of Kv3 positive modulators, which they had already obtained some data in humans for treatment of deficits in the auditory system. So we knew that these compounds had good oral bioavailability. They crossed the blood-brain barrier. They were safe, etc. So we thought, okay, could we use these compounds to increase Kv3 current to address the underlying pathomechanism of progressive myoclonus epilepsy type 7. So here, this is what happens when we add the compound to the channel. So you can see it has complex effects. This is an early stage compound called AUT6, and you can see in a dose-dependent manner at relevant voltages, this is at -10, it increases channel conductance. It also produces a left shift in the voltage dependence of activation. So the effects of the compound on the channel are complicated, but we think at relevant voltages it's acting to increase current. So we tested it on granule cells and cerebellum from 6-month-old, in brain slices from 6-month-old progressive myoclonus epilepsy type 7 mice. The compound increases firing frequency, so in gray, of wild type neurons too. So we actually don't really know if it's acting on mutant channels or on the residual wild type channels, because it exists in a heterozygous state. And actually it's even more complicated because the Kv3 channels in the brain are heteromultimers of Kv3.1, 3.2, and 3.3, and depending upon the brain area. But what you can see is in red is that when we add this compound to the mutant granule cells, it recovers their firing frequency to control levels, and it has no effect on neurons, in this case pyramidal cells, in the neocortex that we know don't have Kv3 channels. So we think it shows some cell type specificity, and it also improves performance on the rotarod, as well as on the ledge test, and then it partially normalizes their accelerated PTZ kindling. So here we gave a single dose of PTZ every 2 days until generalized tonic-clonic seizure. And then in the treatment group, they received an IP AUT6 30 minutes before receiving PTZ. And you can see that it normalizes accelerated PTZ kindling. So, we did long-term video EEG monitoring in 9-month-old mice, and it reduces their spontaneous seizure frequency. So this data was used as to support an IND to the United States Food and Drug Administration, which granted it orphan drug status, and we performed a small clinical trial at the University of Pennsylvania in 6 individuals who were age 21 and older, and we're analyzing the data and should have that hopefully for release maybe by the end of the year or early next year. So just to summarize this part of my talk, we made a preclinical mouse model of progressive myoclonus epilepsy type 7, and it, as expected, shows impaired excitability of Kv3.1 expressing neurons throughout the brain, progressive cerebellar dysfunction, and epilepsy. And this compound, this test compound, and now a more potent compound, enhances Kv3 current and partially normalizes cerebellar dysfunction in vitro, as well as motor dysfunction and epilepsy endophenotypes in a dose-dependent manner in vivo, and this approach may represent a promising treatment for progressive myoclonus epilepsy type 7, maybe other KCNC1-related disorders, and maybe other progressive myoclonus epilepsies. Now I wanted to talk about more about the KCNC1 spectrum. So as Stephanie mentioned, we characterize this valine 434 leucine variant, as well as and defined a new clinical entity in this KCNC1 space, which we argued causes a quote-unquote gain of channel function. So these variants are associated with developmental delay, intellectual disability, central hypotonia, but no myoclonus, and either rare later onset epilepsy or no epilepsy. So it's really a developmental delay, intellectual disability syndrome, and the effects of these particular variants on the function of the channel are gain of function in the sense that this left shift in the voltage dependence are going to create more potassium channel flow at each given voltage. So what's common about these three residues is that they're located between the selectivity filter of the channel and the conserved PXP motif in S6, which is PVP and Kv3 channels, which forms a kink that has been shown to be important for potassium channel gating. And these variants could decrease the stability of the closed state in favor channel opening. So, and that insight was provided by the cryo-EM structure that I mentioned. So this is another case. This was a child who was born to term. There were mild developmental delays were apparent by in the first year of life, but the child walked. He spoke in short phrases, and then he had epilepsy onset at age 1 year with multiple seizure types, including myoclonic seizures, generalized tonic-clonic seizures, but the epilepsy was eventually well controlled. So this is a little bit more severe than in some ways than the previous syndrome that I described, but not as severe as really a developmental and epileptic encephalopathy. Had a normal MRI of the brain, and had this particular recurrent variant, this threonine 399 methionine variant, which again is located in this extracellular S5 S6 linker. So we recorded this variant, which has subsequently been recorded before, and it had absolutely no current. So there's a disconnect here. So we see a similar amount of current to the alanine 421 valine, but the phenotype isn't as severe. It has this sort of intermediate phenotype, but complete loss of function. So this didn't make sense to us. So what we did is we co-expressed this variant with wild type to mimic the heterozygous state of the channel in patients, and we recovered the current. So which now has this similar left shift in the voltage dependence of activation. So we actually are calling this dominant positive. So the channel is non-trafficking by itself, but when you co-express it with wild type channels, it traffics to the membrane and forms heteromultimers at the cell surface with wild type Kv3.1, but causes this left shift in the voltage dependence of activation, similar to what I showed you before. So that's explains why we see this very severe loss of function just with the expression of the homomeric variant, but the clinical phenotype more resembles this sort of intermediate severity. So this is a little foray into a related gene, KCNC2, which is also a disease gene, and it's electrophysiologically identical to KCNC1, but it provides some insight into how these variants cause disease. So I'll just go into it very quickly. We identified this particular variant, this Kv3.2 cysteine 125 tyrosine variant, which again produces this large left shift in the voltage dependence of activation. Here we were sort of interested in this one. Here's the location of the variant, and it's not anywhere near any of the variants that I already showed you. Okay, it's in this T1 domain, the tetramerization domain, which is important for these Kv3 subunits to form these tetrameric structures, which are important for channel function. So how exactly is that happening? Well, we collaborated with Manuel Covarrubias and Lucy Delamotte. This has been subsequently published in PNAS, by the way. But basically, we conduct these molecular dynamic simulations using the cryo-EM structure, and what it shows is something that you would never predict from just the linear amino acid sequence, which is that this cysteine 125 residue in the T1 domain actually interacts with the channel gate to stabilize the open state. Okay, so this is in Kv3.1. The C78 and Kv3.1 is equivalent to C125 and Kv3.2. And look, it wraps all the way around and it interacts with key residues in the gate to stabilize the open state, which leads to this left shift in the voltage dependence of activation. So we developed this for another project, but we have a multi-compartment model of a fast spiking parvalbumin interneuron, and I definitely don't have time to go into this. But what's amazing about the modeling is that we can take these complex biophysical effects of the variant and then isolate them and look at figure out which is the most important biophysical change that drives pathology. So first of all, the major effect is these are input-output curves. So even though we have a quote-unquote, this is an important concept, we have a gain of function of potassium channel, the effect on the fast spiking interneuron is to impair excitability, because as I showed you, that very depolarized voltage dependence of activation is critical for high frequency firing. So when we have a 30 millivolt left shift in the voltage dependence of activation, that's going to impair high frequency firing of these fast spiking interneurons, which is going to lead to less, this is an oversimplification of course, less inhibition in the brain, and then features of intellectual disability and epilepsy. And basically what we can manipulate one parameter at a time because the variant has many effects on channel function, and show that it in fact is this voltage dependence of activation that's the most important feature driving channel dysfunction, which might be important for informing development of novel drugs. So mainly for the community, I just wanted to point out, we're trying to study every KCNC1 variant. So we talked about progressive myoclonus epilepsy. We talked about this gain of function variants, which are defined by developmental delay, intellectual disability, possibly with epilepsy and ataxia. And then we have this developmental and epileptic encephalopathy phenotype. So we made a mouse model of this too. This was tricky, so we made this in a Cre-dependent fashion because we were worried that we would have a very severely affected mouse, which in fact we did. So the expression of the variant is under Cre-mediated control. So in the absence of Cre recombinase, we have expression of the wild type allele, and then in the presence of Cre, we have excision of this duplicated exons 2 through 4, and then read through an expression of this exon 2 with this valine inserted at position 421. And then we cross this to a reporter strain where these parvalbumin interneurons are labeled with TD tomato. Typically you would use a parvalbumin Cre driver line, but we can't do that because then we would only have expression of the variant in parvalbumin cells, which by the way produces a very similar phenotype to the global anyway. But we cross this to a beta-actin Cre, which expresses high-level Cre recombinase from the blastocyst stage. So now we can compare wild-type mice that express Cre and where the parvalbumin neurons are labeled to mice expressing this single amino acid change associated with developmental and epileptic encephalopathy. And these mice are smaller. They have epilepsy. They have this hind limb clasping phenotype, and they have accelerated mortality again from the severe epilepsy. So it's an excellent phenotype of the human condition, and they have a very profound impairment in so this is a brain slice. Here's the primary sensory motor cortex in an acute brain slice. This is work done by Eric Wengert, who's a very talented postdoctoral fellow in my laboratory, and Melody, who's a senior undergraduate at University of Pennsylvania in Philadelphia, Pennsylvania, in the United States. So they make these brain slices, and they can target these parvalbumin cells because they're fluorescent red. They can inject cards into them, and you can see that this I've recorded from fast spiking interneurons in probably, I don't know, 20 different mouse models of different genetic disorders, and these are the most profoundly impaired fast spiking interneurons I've ever seen. So we're using this model, which is an excellent model of this developmental and epileptic encephalopathy phenotype, and we're trying to approach this both with an antisense oligonucleotide-based approach as well as with base editing, which is a CRISPR-based technology to correct single base pairs. So that I apologize for going a little bit over, but I have much more that I could have spoken about, but I wanted to try to cover the entire or as much of the KCNC1 related spectrum as I could. So just to conclude, in general, advanced neurogenetic testing can identify the cause of pediatric neurologic disease, and it's more than explanatory. There are emerging opportunities for that to alter medical management, but we still have a long way to go. We have an incomplete understanding of how genetic variation leads to epilepsy, even for something like Dravet syndrome, or something like KCNC1 that my lab has been studying very intensely for a number of years now. So KCNC1, which encodes Kv3.1, which I've been studying for over 20 years, is an emerging cause of a complex spectrum of neurologic disease, from developmental delay, intellectual disability, myoclonus, progressive myoclonus epilepsy, and then developmental and epileptic encephalopathy. And this data that I showed, as well as other data that we'll see, I think has important implications for various efforts to target Kv3 channels to treat these types of disorders, to try to improve the lives of affected children and their families. So just wanted to acknowledge this is our epilepsy neurogenetics program. To big group. And then, and I just wanted in particular to highlight Natalie Ginn, who's the genetic counselor that I work with in clinic. And then here's the basic science laboratory. And in particular, I wanted to highlight the work of Jade Feng, who was a postdoc in my lab who led the progressive myoclonus epilepsy work. And she now actually works for the FDA. Eric Wengert, who's studying the alanine 421 valine variant, and his paper should be on bioRxiv hopefully by the end of the month if anyone is interested. And then I mentioned Jerome Clateau, who's an expert ion channel physiologist in the lab, research scientist who did all the hex cell work. And key collaborators on the Progressive Myoclonus Epilepsy Project were Nayara Akizu at CHOP, and then other collaborators, of course, are Manuel Covarrubias at Jefferson, also in Philadelphia, Lucy Delamotte, our collaborators at Tiffany, and then Tim Vogels on the computational side.
Thank you very much, Dr. Goldberg, for that excellent presentation. It really did capture the KCNC1 story from patient to lab to clinic. We really appreciate that. Dr. Gary Clark, the Director of Screening Technologies. He has over 20 years of ion channel drug discovery experience with 14 years in the contract research arena. I've had the pleasure of knowing Gary for most of that time. Kind of grew up together in the ion channel world. Gary's interest in ion channel started during his degree at Royal Holloway University of London, where he performed sharp electrode recordings with leach ganglia, which led him to a PhD in cardiac electrophysiology at Coventry University. Following his PhD, he worked at several biotech companies where he was closely involved in the early development of automated patch clamp technologies. That's where we overlapped. Then he moved on to take a team leader position at Biofocus, which became Charles River in 2005, where he implemented and established automated electrophysiology for their contract research services. So Gary's presentation will be entitled A Fluorescence-Based Drug Repurposing Screen of the Potassium Channel Kv3.1 with V434L mutation.
Okay, thanks, Chris, for that introduction. Also, I'd like to say big thank you to Dr. Goldberg for his presentation as well. That was really enlightening. And also a big thank you to Stephanie as well, who I will say, I've really enjoyed and working with her on this project. It's probably been the most humbling project I've done in my career. And I hope that we can help her, her daughter Eliana, and also other people's children as well with the work that we have done with her. So as Chris mentioned, at Metrion, we embarked on a project with Stephanie and the team at Perlara to screen the drug repurposing library from the Broad Institute against the Kv3.1 channel with the V434L variant. Oh, let me just click there. So obviously Metrion Biosciences is a specialist contract research organization that provides ion channel drug discovery services to the pharmaceutical and other bioscience customers worldwide. I won't dwell on all the things that we can do because Chris has mentioned that earlier in the at the start of the webinar, but obviously there's a whole host of information on our website that you can look at as well. So I'm going to just delve right into the purpose of the project that we had with the KCNC1 Foundation, and that really was to identify our antagonists of the Kv3.1 variant that Eliana had from the Broad repurposed drug collection, with the idea of finding a potential new therapy for Eliana. And the reason for this was so that we could find something as quickly as possible so that these potential treatments could be given to Eliana as quick as possible. Now, the overview of our project was really to confirm the functional expression of the Kv3.1 channel, both the wild type and also Eliana's variant in a recombinant expression system. And then once we had done that, we then created a stable cell line that was going to be robust enough for us to then use in a fluorescence assay, which we could then use on our Flipper Penta to then screen the Broad library. And then following any hits that we obtained from the primary screen, we would then look at the potency of those hits that came through. So this slide really just replicates pretty much the work that Dr. Goldberg's team had done in his paper, and we were really pleased that we were able to reproduce the same sort of data that he had published. And again, you can see, like he had demonstrated, there was a leftward shift in the voltage for activation in the KCNC1 variant. And when you looked at the tail current amplitudes, you could see that there was always a persistent inward current at hyperpolarized voltages, which, as Dr. Goldberg mentioned, was then had the effect of hyperpolarizing the cells, and then was dampening any excitability of those cells. And obviously you can see the current traces on here with the wild type channel versus the variant channel, and then obviously cells that were non-transfected, you can see there was very little current, if not any. And it was important for us then to do some rudimentary pharmacology just to see if there's any differences in the two different variants of the channel, so wild type and the variant. And we were surprised to come across that when we looked at just two modulators, obviously we've talked about the orthophenol compounds in the previous presentation, that we found some differences between the wild type and the variant. So you can see on the left hand side we can demonstrate a nice concentration dependent effect of a broad spectrum potassium channel blocker called 4AP. However, with Eliana's variant, we failed to inhibit that completely, even at quite heroic concentrations. And perhaps more surprising was when we applied the Kv3.1 activator ORT1 that was published and commercially available. We saw activation of the wild type Kv3.1. However, we saw inhibition of Eliana's variant, and so that made the important decision for us that we would need to screen the Kv3.1 variant into looking for compounds that would inhibit Eliana's variant as possible therapies for her going forward. So we initiated a cell line development by stably expressing the gene of choice for the variant, and we screened over 190 monoclones using a limiting dilution cloning exercise, and those clones were then tested on our Flipper Penta using a thallium flux assay. And you can see down here in the bottom left-hand corner the different responses we saw with the activation of the various different clones. So each one of these wells would have been a single clone or in duplicate. So you can see actually when we add a higher potassium concentration to the wells where we depolarize the cells, we can then see a thallium influx into the cells, which lights up and causes a fluorescence effect. So you can see here on the right-hand side, the negative clones, there appears to be little or no thallium influx from fluorescence, but obviously some of these putative clones, we saw a large fluorescence due to the thallium influx. So we took 4 of these clones and then tested them on our Sapphire Cube automated electrophysiology platform. And here you can see the 384 well view of the plate, where we have put the 4 different clones in rows of 4. And what you're seeing on this screen here is the individual current activation or the current voltage plot as we did an IV for this. So, and we were pleased to see that actually the majority of the clones had, or 3 of the clones had very good expression. So the top clone in 1A9 had a close to 60% of the individual cells showing a Kv3.1 expression. However, our favored clone was clone 2G6, where we saw close to 100% of the recordings with a current resembling Kv3.1. Now, as part of that, obviously we have QC criteria where we ensure that the seal resistance of the cells were greater than 200 megaohms, and anything we deemed to show expression of Kv3.1 had greater than 400 picoamps worth of current. So with our desired clone that we had identified from the Flipper and then further confirmed on our Cube system, we then set about developing a fluorescence assay on the Flipper that was then going to be both accurate and robust for performing a screen at a single concentration. So there had been a paper whilst we were developing this assay where they showed that fluoxetine had helped, was being trialed as a possible, showed improvements with fluoxetine in another patient with a gain of function variant. So we included fluoxetine as it was easily available on our plate for testing on the Flipper. So this was a schematic of a typical Flipper plate where we have our TEA full block controls and our DMSO controls in the columns 1, 2, 23 and 24. And also we did a concentration response curve for both ALT1 and fluoxetine, and we were able to, using our instrumentation, randomize where those different concentrations were placed onto the plate, so that when we could then assess the robustness of the assay. So this slide really just shows the 2 experiments we've done on plate 1 and plate 2. They were identical in where the compounds were applied, and you can see we have our full block TEA controls giving 100% inhibition, and then in the vehicle controls of 0.5% DMSO, you can see in blue, so hovering around the 0 line, and then blank wells, they were the wells in the middle, in the columns 3 to 21 that didn't have any compound in, and then the black and pink dots highlight the different concentrations of ALT1 or fluoxetine that were tested on the plate. So you can see varying degrees of inhibition for the different compounds there. And when we pulled the data for ALT1 and fluoxetine together and constructed their relevant concentration and then inhibition, we were able to construct concentration response curves that were very robust and tight, and also consistent from plate 1 to plate 2. So the decision was then to, as the assay was deemed robust and fit for purpose, we then obtained, with the help from the team at Perlara, they negotiated with the Broad Institute to supply their repurposing library as assay-ready plates. And we screened just over 6,700 compounds in duplicate. And you can see on the left-hand side some representative traces of compounds that we observed. So compounds, so the traces in 2, 3, and 4 were compounds that were deemed to probably be inactive. So you can see the green traces are full response activation of the channel, and then obviously the blue traces here indicate the effect of full or the concentration of TEA that was used to block the channel, and then we're getting different levels of fluorescence in between control and full block. Obviously, compound number 7 or trace number 7 appeared to inhibit further than the TEA that we had on the plate, and then trace 1 will obviously appear to show activation of the channel as well, or increased activity of the channel. So there's just over 45 plates that were screened in total for both the duplicate 1 and duplicate 2, and you can see that the plate statistics of the mini screen were very reliable, with our Z prime or robust Z primes being greater than 0.6, and the mean Z prime was actually 0.72, and we had a good signal window of greater than 3 in the plates that were screened. This slide just shows the duplicate plates from run 1. So this is all the plates, individual plates screened on the first occasion, and then the same plates, or the same compound plates, repeated in the second occasion. And you can see that this is just a heat map showing activity, where the darker the red indicates the greater the inhibition, and then the cooler the color in blue shows that we're seeing activation. And what you'll see is the big stripes going down the sides of the plate, obviously indicating the TEA chloride, and then the white is obviously the DMSO controls. And you can see that the same sort of patterns are being picked up in the same plate in run 1 compared to run 2. So if we take this plate here, which is 190, you can see we've got a kind of a hot spot here in the middle, and again the same plate that was the same compound plate that was tested on another occasion again shows the same sort of pattern. And so when we pull all that data together and correlate the test 1 or against test 2, we can see that we get a very good correlation of the inhibition data, or the actual percent of control data. So what we've done here is obviously we have correlated the inhibition or the percent of control data, and also color coded it with the class of compound based on the what was in the Broad library. So you can see that compounds that dots that are colored in blue are compounds that have been launched and prescribed to patients. And obviously that was important to carry through as many compounds as possible that were already on the market, so they could be considered as being prescribed to Eliana. And as I mentioned before, this is percent of control data, so we are normalizing the data to the TEA condition, and obviously some compounds we observed greater inhibition, and that's not necessarily a surprise, as TEA is not the most effective inhibitor for this particular channel. So from this initial screen, we then took 80 compounds to then be determined their potency. For this set of part of the project, these 80 compounds were tested as 10-point concentration response curves from a top concentration of 30 micromolar, and there were half-log dilutions going down. So there were 6 plates tested in total with 14 compounds per plate, and obviously you can see here that of those 6 plates that were tested, we had good robust Z prime values of 0.68, so comparable with what we saw on the screen. And again, we kept on to the 2 tall compounds to demonstrate that we are seeing consistent pharmacology from plate to plate. So clearly we had a high hit rate from the initial primary screen at a single concentration, but we also got very good confirmation of those hits when we took them to potency determination. So of the 80 compounds, there were 12 where we demonstrated less than 1 micromolar of an IC50. The majority of the compounds had IC50s ranging between 1 and 5 micromolar, and then 18 were had an IC50 greater than 5 micromolar. And here just on the left hand side is an example of one particular compound that gave an IC50 of 18 nanomolar. So that's where we are with the project, and obviously the KCNC1 Foundation has all this data, and they are then using it with their consultants to decide how to use this information then to go forward as a possible therapy for Eliana. But just to sum up on where we are, we obviously confirmed that that the V434L mutation in the KCNC1 gene did create a gain of function Kv3.1 channel, and we then went on to generate a cell line which we then were able to use on the flipper and also later on the cube. We developed that assay on the flipper so that we could test compounds confidently at a single concentration, and whilst we tested just over 6,700 compounds, we identified 80 hits from that phase to then take on to potency, where we saw IC50s of less than 5 micromolar in the majority of the compounds. So with that, I would just like to acknowledge various people who were involved in this project. So that's Scott, Emilio, Tom, Graham, Matt, and obviously all the cell culture team that are involved in keeping the cells in culture all the time. I'd also like to, in particular, to thank Stephanie at the KCNC1 Foundation and the team at Perlara that we work with closely as well.
Thank you so much, Gary. It was definitely worth it. And we are going to take some time for Q&A. So we'll start with Gary. A question for Gary from the audience. It says, since you have a lot of experience with automated patch clamp, can you discuss the pluses and minuses of doing the fluorescence-based assay versus doing the characterization with automated patch clamp, where you could get more detailed information on channel gating, et cetera?
Yes. So yeah, obviously Metrion, we have a huge amount of experience in automated patch clamp for HTS and also fluorescence-based systems. I think the general theme is that a fluorescence-based assay will give you a kind of a yes/no answer on hits. It's generally faster and more cost-effective to screen on a flipper or a fluorescence-based assay. However, it doesn't give you the same kind of level of detail or information that you will get on a sort of an automated platform.
Thanks, Gary. So here's a question for Dr. Goldberg. With the Tiffany compound being tested in a clinical setting, What are the next steps in the process for this compound? Will this compound be used to treat other conditions in order to reach a bigger target market?
Yeah, thanks for the question. So I don't entirely know the answer to that, so we're still analyzing the data, and there are a range of compounds with different properties, and which compound or compounds might come to patients and which patients might be eligible, I think remains to be seen. But there is data that these compounds are safe, and we're hopeful that uh, that preclinical program will lead to something that can be available to patients at some point. But that data still has to be uh, analysis of the data still has to be completed. And I should just mention that the clinical trial was run by it was an industry sponsored trial that was run by a colleague of mine at Penn. And I'm not a clinical trialist, so I wasn't directly involved in the clinical trial.
Okay, thank you. There's a question about fluoxetine, which is Prozac, an antidepressant. Gary showed some data in his presentation. Dr. Goldberg, is that something that's being used as a treatment in clinical settings for these disorders?
It's a very complicated question. So
Yeah,
we have tried fluoxetine in one patient. I think that there are millions of people who've taken fluoxetine, and
right.
if it acted as a Kv3 channel modulator, uh, I wonder what its action might be on
Mhm.
native Kv3 channels in the brain. I think more work sort of needs to be done on that. Uh, so we've had limited experience with it so far. But
Okay.
as Gary mentioned, there is a published case report with some data in heterologous systems, and then some sort of correlative clinical data. This is always an issue with these N of one case studies where let's say you have a child with a neurodevelopmental disorder that has been ongoing for many years. They're already on many anti-seizure medications. They receive another trial medication for some period of time, and interpreting what the effects of that medication are and whether they can be attributed to that particular medication trial is very, very complicated.
Yes, understood. And it's kind of a related question. I'm sure people are wondering, have any of the compounds identified by the Metrion screen been used to treat Eliana at this point? Have they been effective? I know it's a recent study, and so I know it's ongoing, but have there been any developments in that area?
So we're just finishing a trial with fluoxetine based on the case report, not necessarily from the results from Metrion, but we will be starting an N of one trial for Eliana probably sometime this fall with one of the hits from the drug study. So more to come.
Excellent, thanks. And speaking of that, I was wondering, how did you find Metrion? How do you educate yourself in this process? Is it just Google searches? Is it simple as that, or word of mouth? How did you do that?
Great question. I came across Perlara that helped me identify Metrion, and Perlara is kind of a group of PhD scientists who are helping families or small foundations like mine navigate the space of um, funding a treatment for their child. So they helped me identify Metrion as being a leader in ion channel drug discovery. Um, and so, based on their word of mouth and discussions with individuals in the space, they us recommended me looking into Metrion, and we went that way. So, we were able to sort of look at all the different CROs out there, and this one just seemed to be the best fit.
Excellent, thanks. Here's a question from the audience for you, Stephanie. It says, thank you for your work with the KCNC1 Foundation. I know you're working on a ton of fundraising opportunities, which is amazing. What are the next steps in the foundation's growth?
It's a great question. So, there's we're trying to work on different steps. So I think our next major step is to create a patient registry and to get that off the ground. I think a lot of the patients on our online community are really keen to participate and to share their information and their child's progress. So I think that will be my next big focus for the foundation. And in terms of, steps for a treatment for other individuals, I think that is going to take a little bit more time and more fundraising. It's a big endeavor for a very small foundation. But
right.
Yeah, hopefully with collaboration and just working with others, we will be able to expand into different variants, and even looking at the other gain of function variants to see if any of the results from our current study on Eliana's variant is relevant for that group.
Excellent, thanks. Yeah, I think the registry is a very important step. So we'll do one more question for Dr. Goldberg. In this presentation and your presentation, Gary's, there was a lot of amazing technologies used: cryo-EM, automated patch clamp, even manual patch clamp electrophysiology. What do you see as a really important next step in the technology realm? AI is really popular these days. There's gene therapy. What do you think is the next big advancement for this kind of research?
Well, it's always difficult to predict the potential impact of technologies that are under development or haven't been developed yet. But I think actually, if I could sort of change the question a little bit,
Sure.
I think an important next advance is understanding how disease-associated genetic variants interact with development. So we know that, for example, KCNC1-related disorders are due to de novo, or in some cases, inherited variants that are present from conception. But just like the healthy human brain, the brain of a child with a neurodevelopmental disorder also continues to develop, maybe in a different way. And that may be partially compensatory or corrective. It may be part of the pathologic process, or it may be epiphenomenal, such that the actual status of a child at age 1, 2, 5, 10 involves the underlying genetic variant plus all of these other processes, such that the actual target might be changing and might not be directly informed by the biophysical effects of the variant on the channel as it appears in, for example, a heterologous system. So I think maybe not a new technology itself, but a greater understanding of what these technologies are telling us and linking across them, I think is important. But if I could just add something that I do think would be very helpful, would be improved ways of understanding the subunit composition and subcellular expression of Kv3 channels throughout the brain. So we still don't really know what is the native state of these channels. They heteromultimerize, they're modulated by second messenger systems, etc. They have very complex patterns of expression in specific cell types in different subcellular compartments. And I think understanding that is something that, again, some of these single cell biology or high resolution imaging technologies maybe could tell us in the future.
Excellent. Thank you very much for that comment, and thanks everyone for the great questions. Thanks again to Stephanie, Dr. Goldberg, Dr. Clark for providing excellent presentations. And so we have